this episode, we are going to see about the antibacterial expression system. As we go ahead with this antibacterial gene expression system, first we are going to see about the types of the immunity an organism can get. Innate immunity provides an organism with the first line of host defense against pathogenic organisms. As opposed to adaptive immunity, this response is non-clonal, non-specific and ever-present. In addition to its constitutive quality, there is a rapid response whereby components of the innate immunity are produced after an initial challenge of pathogens. Among the cells that make up the arsenal of the innate immunity are circulating phagocytic cells such as neutrophils and macrophages, in addition to the cells that line the mucosal epithelium. One common characteristic of cells from these two distinct lineages is the production of antibacterial and antimicrobial peptides, which act as a part of the armamentarium of the cells to prevent microbial colonization. The peptides in the phagocytic cells act as a part of oxygen-independent microbicidal pathway, whereas in epithelial cells, they are presumed to be secreted into the extracellular environment to provide antimicrobial activity. In both cases, the genes that encode these peptides can either be constitutively expressed or induced by inflammatory mediators and bacterial challenge. Antimicrobial peptides or proteins, also called as AMPs, are a group of immune proteins that exhibit strong antibiotic properties against numerous infections, bacterial strains. They are evolutionarily conserved and present in every kingdom and phylum ranging from prokaryotes to humans. Antimicrobial peptides, also called host defense peptides, are part of innate immune response found among all classes of life. Fundamental differences exist between prokaryotic and eukaryotic cells that may represent targets for antimicrobial peptides. These peptides are potent, broad-spectrum antibiotics which demonstrate potential as novel therapeutic agents. Antimicrobial peptides have been demonstrated to kill gram-negative and gram-positive bacteria, envelop viruses, fungi, and even transform or cancerous cells. Antimicrobial peptides may also have the ability to enhance the immunity by functioning as immunomodulators. Antimicrobial peptides can be divided into numerous categories based on their primary and secondary structures, but most of them maintain certain common structural characteristics. These include a cationic charge due to arginine and lysine residues and a greater ability to interact with bacterial membranes due to an overrepresentation of hydrophobic amino acids. The peptides also exhibit a broad spectrum of antimicrobial activity, often including both gram-positive and gram-negative bacteria, fungi, and envelope viruses. In addition, antimicrobial peptides are frequently found in families where a single species may have numerous homologs expressed in the same cell type, suggesting complementary or synergistic activities between homologs. Lower organisms such as Amphibians and fish express a variety of linear antimicrobial peptides in their skin secretions. These exhibit an amphipathic alpha helical structure and are similar to insect peptides which are abundantly expressed in the hemolymph. Antimicrobial peptides are a unique and diverse group of molecules which are divided into subgroups on the basis of their amino acid composition and structure. Antimicrobial peptides are generally between 15 and 50 amino acids. These peptides include 2 or more positively charged residues provided by arginine, lysine, or in acidic environments, histidine, and a large proportion, generally more than 50%, of hydrophobic residues. The secondary structures of these molecules follow 4 themes, including 1 stage, it is alpha helical; second stage, beta stranded due to the presence of 2 or more disulfide bonds; at third stage, beta hairpin or loop due to the presence of single disulfide bond and cyclization of the peptide chain, and at 4 it is the extended sheets. Many of these peptides are unstructured in free solution and folded into their final configuration upon partitioning into biological membranes. It contains hydrophilic amino acid residues aligned along one side and hydrophobic amino acid residues aligned along the opposite side of the helical molecule. This amphipathicity of the antimicrobial peptides allow them to partition into membrane lipid bilayer. The ability to associate with membranes is a definitive feature of antimicrobial peptides, although membrane permeabilization is not necessary. These peptides have a variety of antimicrobial activities ranging from membrane permeabilization to action on a range of cytoplasmic targets. Insects are continuously exposed to potentially pathogenic microorganisms and eukaryotic parasites, but only a few encounters results in infection. Insects possess a complex and efficient system of biological defense against pathogens and parasites. This involves the following: A. The integument and gut as physical barrier to infection. Number 2. Activation of prophenol oxidase cascade and coordinated responses of several subpopulations of the hemocytes when these barriers are breached. And next, the induced synthesis of antimicrobial proteins primarily by the fat body and their synthesis induced within hours following a septic injury. Vertebrates have the acquired immunity with immunological memory, whereas invertebrates lack this immune system, but instead they possess an innate immunity which is characterized by non-specific immune reactions against foreign materials. In general, the insect immunity consists of cellular and humoral reactions. Cellular reactions involve phagocytosis, nodule formation, and encapsulation by the plasmocytes and granulocytes. On the other hand, humoral reactions involve activation of the prophenol oxidase cascade and induction of immune protein such as lysozymes, lectins, antibacterial proteins, and antifungal proteins. Both immune reactions work in concert to prevent insects from acquiring infections with microorganisms. The injection of bacteria into pupae of the mouth resulted in the synthesis of immune proteins. These proteins are purified and characterize novel classes of antibacterial proteins which are called as secropins and atacins. Antibacterial proteins are known to be rapidly induced mainly in the fan body and hemocytes upon the bacterial infection or a wounding. Insect antibacterial proteins are heat stable and have a broad antibacterial spectrum. Many antibacterial proteins have been isolated from various insects, and at least 4 types of secropin, atacin, lebosin and moricin are found. Secropin consists of about 40 amino acid residues with 4 kilodalton molecular weight and is heat stable. 3 subtypes of secropin are available. The secropin A, secropin B and D have been reported from Bombyx mori. Secropin contain 2 alpha helices and are rich in basic amino acid residues at the N terminus and in hydrophobic amino acid residues at the C terminus, which is presented. Secropins are active mainly against gram-negative bacteria. It was demonstrated that secropin forms ion channels in the bacterial membrane and as a result bacteria are killed. Atacin is glycine rich and has a molecular mass of 20 kilodaltons. Atacin acts against growing gram-negative bacteria and was shown to inhibit synthesis of the bacterial outer membrane. Lebosin consists of 32 amino acid residues and isoproline rich. An amino acid residue of lebosin, which is the threonine at the 15th position, is glycosylated, and this sugar is important for expression of antibacterial activity. The primary structure and the antibacterial character of the lebosin resembles those of abscin, around 41% identity in the amino acid sequence, which is an antibacterial protein of the honeybee, although abscin is non-O-glycosylated. 3 lebosin analogues, lebosin 1, lebosin 2 and 3 have been isolated, and it was found that lebosin 1 and 2 have common amino acid sequences which is given in presented. Moreover, the glycosylated threonines of these antibacterial proteins contain different sugars, namely N-acetyl galactosamine and galactose lebosin 1 and N-acetyl galactosamine lebosin 2, which is presented. The amino acid sequence of lebosin 3 shows one amino acid sequence replacement of leucine at 16th position and threonine at 15th position is modified with N-acetyl galactosamine. Recently, a new member of the lebosin gene family called the lebosin 4 has been cloned, and its deduced amino acid sequence reveals a high similarity together with the lebosin members. The amino acid sequences of the antibacterial proteins from insect that is taken from Bombyx mori. Amino acid sequences of atacin and lebomin 4 were deduced from the nucleotide sequences of the cDNA and the gene respectively. The nature of the glycosylation of threonine at 15th position of the lebosin 4 remains unknown. The incubation of lebosins with the liposome preparation causes leakage of entrap glucose under low ionic strength conditions, suggesting that the bacterial membrane is a target for leucine. Since leucines show very weak antibacterial activity under physiological conditions and require low ionic strength for full expression of their activity, their biological significance in the immune mechanisms remain obscure. These complicated features of the leucines suggest that although they have very weak antibacterial activity, they might primarily serve to reduce the minimum inhibitory concentrations of other antibacterial proteins under physiological conditions. Indeed, leucine 3 was shown to have a synergistic effect with sericotropin D, suggesting that antibacterial proteins work cooperatively in the immunity of Bombyx mori. Such a synergistic effect was also observed between an antibacterial protein sarcotoxin 1A and an antifungal protein in a flesh fly Sarcophaga peregrina. Another novel antibacterial protein designated morisin was isolated from the hemolymph of Bombyx mori, and it showed antibacterial activity against Staphylococcus. This protein consists of 42 amino acid residues and is highly basic. It had no significant similarity with other antibacterial protein. Morisin has antibacterial activity against several gram-negative and positive bacteria, has a higher activity against gram-positive bacteria than a sericotropin B and is inducible upon bacterial injection. These results suggest that the protein is responsible for antibacterial activity against gram-positive bacteria in a Bombyx mori. The effects of the protein on bacterial liposomal membranes indicate the target of the protein is the bacterial cytoplasmic membrane. The results also suggest that the N-terminal portion of this protein containing a predicted alpha helix is responsible for an increase in the membrane permeability. The results also suggest that the N-terminal portion of this protein containing an predicted alpha helix is responsible for an increase in the membrane permeability. The expression of bacterial resistance to antibiotics is frequently regulated, which indicates that modulation of the gene expression probably reflects a good compromise between energy saving and adjustment to a rapidly evolving environment. Modulation of gene expression can occur at the transcriptional or translational level following mutations or the movement of the mobile genetic elements and may involve induction by the antibiotic. In the latter case, the antibiotic can have a triple activity as an antibacterial agent, as an inducer of the resistance to itself, and as an inducer of the dissemination of the resistance determinants. The lysozyme gene is constitutively expressed in insects at a low level in the fat body, hemocytes, and epidermis cells and is strongly expressed upon bacterial infection. In addition, lysozyme is considered to contribute significantly to the production of soluble peptidoglycan PG, a strong inducer of the antibacterial proteins. 2 lectins have been reported from Bombyx mori. One has a molecular mass of 260 kilodaltons and another designated hemocytin, the molecular mass of 280 kilodaltons. This hemocytin is regulated to a mammalian coagulation factor, also called as von Willebrand factor. The characteristic feature of the carbohydrate recognition domain of C-type animal lectins were detected at the C-terminal sequence of the hemocytin. When cDNA encoding this region was introduced into a baculovirus vector, hemagglutinin activities were detected in the culture fluid of the recombinant virus-specific cells. These activities were inhibited by D-mannose, N-acetyl D-galactosamine and D-maltose, which are heptanic saccharides of authentic hemocytin. The hemocytin gene is transcribed in hemocytes of the insects at the larval pupal metamorphosis and or after injection of E. coli and lipopolysaccharides LPS. The hemocytin plays an important role in both immunity and in metamorphosis. Hemocytin also has a homologous region with coagulation factor 5 and 8. The regulatory motifs essential for the antibacterial gene expression are as follows. The inducible and tissue-specific regulation of immune genes expression in the insects seems to be at the level of transcriptional induction. To understand the mechanisms underlying this control, it is crucial to identify the cis-regulatory elements involved, and such studies have been initiated by several groups. Knowledge of transcriptional control of immunoglobulins and acute phase genes in mammals led to the important discovery of the striking conservation among some of the insect antibacterial protein genes of a sequence motif with homology to the mammalian KB motif. This binding site was first identified in the enhancer region of the immunoglobulin light kappa gene and has later been found in promoter and enhancer elements in many mammalian genes that are involved in immune, inflammatory and acute phase responses. The KB motif has subsequently been identified in the promoter region of most of the inducible immune genes of insects, including Bombyx mori. The silkworm represents the location and orientation of some putative regulatory motifs in the upstream region of the Bombyx mori, cercropin B1 gene. Numbers given there refers to the distances from the transcription starting site and the translation starting site is indicated by ATG. The white arrows indicates KB motif and the black arrows indicate the GATA motif, the GATA motif. The KB-like sequence present in the 5' upstream region of the insect's antibacterial gene is presented in table 1. The table 1 represents the KB-like sequence present in the 5' upstream region of the insects taken from Bombyx mori antibacterial protein genes. Position of the nucleotide sequence is calculated from the starting site of the transcription as +1, and in the case of leucine, it is calculated from the beginning site of the translation as +1. Comparison of KB sites in the inducible immune genes of insects from the Bombyx mori led to the definition of a consensus sequence for this motif, which is as follows: GGG, RA, NNNN and Y. Transcription factors belonging to the REL family were involved in this gene activation mechanism in Drosophila melanogaster and Helicoverpa cecropia. Of these factors, dorsal related immune factors, the DIF, was identified in Drosophila melanogaster, and DIF was demonstrated to bind to the KB sequence in the 5' upstream of the antibacterial protein in genes. DIF is known to be retained in the cytoplasm by cactus under non-infectious conditions, but it is dissociated from the cactus and moves into nuclei upon bacterial infection. In addition, dorsal involved in the dorsoventral determination in embryogenesis of Drosophila melanogaster is also revealed to activate transcription factors of KB containing a reporter gene. Dorsal gene expression is stimulated in the fat body by bacteria, suggesting that dorsal is involved in the activation of antibacterial protein gene expression. The role of dorsal and DIF in the insect's antibacterial gene activation is unclear. Apart from KB motif, another DNA sequence called the GATA sequence present in the 5' upstream regulatory region plays important role in the induction of antibacterial protein in genes. GATA sequences were also detected in the 5' upstream region of the insect genes encoding secropin A1, secropin A2, secropin B1 and B2, atacin and leucine, which has been presented in Table 2. Table 2 represents the GATA sequence present in the 5' upstream region of the insects, that is Bombyx mori, antibacterial protein genes. Position of the nucleotide sequence is calculated from the starting site of the transcription as +1, and in the case of oflibosine, it is calculated from the beginning site of translation as +1. A consensus sequence of GATA region was WGATAR. A similar type of sequence was also reported in Drosophila melanogaster. It is assumed that GATA sequence is important for insects' immunity. It is also speculated that since the GATA sequence is located in the vicinity of KB sequence, the factors related to these sequence work cooperatively to activate antibacterial protein genes. Recent advances in molecular characterization of innate immunity in insects have revealed striking similarities with mammalian innate immunity, suggesting that signal transduction mechanisms share a common evolutionary ancestry. Due to the enormous use of antibiotics, most of the clinical pathogens are becoming resistant to those antibiotics. Promising alternative antibiotics are the antibacterial peptide antibiotics from insect source, which can kill their specific targets by damaging the bacterial cell membrane.